lecture on genome-wide association studies. So this will be dealt by our most respectful Dr. Samsiddhi Bhattacharjee. He is from NIBMG Kolkata. So he has vast experience in this area of GWAS. So let us look forward. He will share his experience about GWAS as well as whatever practical session, if possible, he will try to conduct. So for the coming 2 hours, it's yours. Please go ahead. Thank
you. So, although I say I will discuss some of the issues, applications of GWAS in general, but my experience has mostly been or almost completely been in human genetics, human GWAS, but I'll try to give perspective or differences between in plants or animals whenever possible, but these are not, I don't have live or practical experience on plants and animals. Okay. So what is so GWAS actually started with human diseases at the beginning. It's meant for mapping binary traits or like common diseases in humans, but of course it can be used for other things like mapping quantitative traits in humans or in plants and livestock. So essentially GWAS is a tool that works for wherever you have polygenicity. For common diseases, there was a hypothesis called common disease, common variant hypothesis. That means, diabetes and even cancer, but not very rare Mendelian kind of diseases. There are many variants across the genome, so that is what it is meant for. But even if you consider quantitative traits like height or IQ and things, BMI in humans, obviously, as you can expect, if you have only one, say, biallelic locus, it is not possible to generate so much of variation in height. So there must be many loci which to put together create this polygenic kind of distribution. So what are the applications of GWAS results, GWAS downstream? That means once we have found out loci which are associated with a trait or a disease, we would be able to point to the genes and the pathways that are involved in that respective trait or disease, and we would be able to target those genes or pathways, therapeutic targets, and we would be able to probably modify them for our benefit. And in humans, something that is very popular nowadays is people are talking about personalized medicine, which is essentially nothing but predicting risks, and also in terms of medicine, which medicine will work, which will not work. This can be done from GWAS, and also this is important area in particularly plants and animals, is prediction of traits, which is just like risk prediction of traits and breeding values for commercial use. So there are two ways of mapping genes, two broad ways. There are some one or two other ways, but genetic linkage and genetic association. Genetic linkage tries to map loci which have sort of limited crossover with the trait loci, whereas association tries to measure correlation between trait and a marker. So in humans, linkage has not turned out to be, apart from those Mendelian completely penetrant diseases, is not very useful, because linkage is low power and because it needs large pedigrees and more number of generations for more number of meiosis. In plants and animals, it is more powerful because you can design your own pedigrees, you can actually breed them. Association mapping works in both humans and plants and animals. It is more powerful, you can get closer to the actual loci. However, there is only one caveat with association mapping, that is because it is not confined to families and things like that, it is more susceptible to population structure. We will come to what I mean by that. Okay, first of all, what is this any questions? Please feel free to stop me anytime. Okay. Any question? No. Okay, so what is this association concept? There are individuals in the population and there are SNPs in the genome, and essentially these SNPs are in what is, there is a phenomenon called LD, which is if SNPs 2, so in this case SNPs 2 and 3 are in LD, because as you can see wherever there is T here, there is G here, wherever there is T here, there is G here, wherever there is A here, there is C here. This pattern you can follow. So that means there is a correlation between the allele you find on a chromosome at this locus and the allele you find on that chromosome at this locus. So the idea of GWAS is that GWAS will only work, you cannot suppose you are not able to type the actual disease variant. GWAS works because if you type close enough SNP that it is in LD with that causal variant, then it will show an indirect association with the phenotype. So this is the gene here, it shows, I think it says unobserved causal SNP. It's kind of hazy, right? Full screen is problematic. Yes, so this is unobserved causal SNP. So this is the unobserved causal SNP, and there is a causal association between this SNP and the phenotype. However, this is SNP may not be measured, but you have an association LD with this genotype SNP. So this SNP will show an indirect association with the phenotype. So this was the original principle of GWAS. So people started developing GWAS chips, which is genotyping chips of common variants. Nowadays it is also being done from sequence data, but this was the original idea. So how are these chips designed? They are designed in this way. This is the chromosome, and SNPs close to each other tend to be in a strong LD. So you sort of select a representative SNP called the tag SNP from each of these chromosomal regions. So the idea is that whatever there are a few possible common haplotypes. So if you type these 3 SNPs here, they will make sure that you have considered most of the haplotypes that are seen in this region. So these are called the tag SNPs, and then they are designed on the chip, GWAS chip, and that they are used for genotyping. Okay, so what is genetic association? It's something like this. You have this particular SNP, these are the 3 genotypes, and if you look at the distribution of the trait, then there will be a trend, the mean of the trait will shift as you go from one genotype to other. You can also see it from this one, the histogram. The red histogram is for capital A genotype, the what color is this, I don't know, gray histogram is for capital A capital G genotype, the heterozygous, and the blue histogram is for capital G capital G. So as you can see, it is gradually shifting to the right. So we want to identify SNPs of this type, and what GWAS does is it goes through all the SNPs in a loop, one by one at a time, and checks for association at all of these SNPs. So in this case, the SNP 1, we are looking at its correlation with the trait, and then we will move gradually to SNP 2, SNP 3, like that. Okay, so I'll sort of skip this. In humans, the First sort of GWAS, successful GWAS appeared in 2007 or so, and within about a decade, just 10 years, this you can see the number, this many unique SNP trait associations have been discovered, and people have made this kind of statements about GWAS. So I'll just show in a graph what it looks like for humans at the moment. This is August 2018. So these are the chromosome 22 chromosomes, and the different color codes stand for different categories of phenotypes that each of these SNPs map to or have been found to be significantly associated through GWAS. This is available, the live, I mean it is real time updated kind of thing. It's available on EBI GWAS catalog. Okay, so what is this problem of population stratification I was talking about? So here we are trying to explain that this is the human context, but it can be in any context actually. So we are looking at a particular quantitative trait that is different between North Indians and South Indians, say due to a lifestyle factor like diet difference. Okay, so but what happens is that we also know this ethnic difference is there. So many SNPs in the genome have different frequencies between North India and South India. Okay, so if we did not know, if we have a sample of people and we completely are ignorant about this origin of the people, their ethnicity, North and South, we just look at our association between the traits and the SNPs, the trait and the SNPs, what will happen is that we will see spurious association for many SNPs in the genome. So whichever SNP you have allele frequency difference between North and South, it will automatically spuriously show association with that trait. Why? Because that trait is different, not for genetics, not for biology, but because of some diet or some lifestyle factor. Okay, so this in this case in epidemiology we call this confounding variable. Population stratification in this case is a confounder, and it needs to be adjusted so that we don't end up with so many false positives in the genome. So this is what we mean by confounding in epidemiology. So in this case ancestry is the confounding factor. So the confounding variable should be associated with both the dependent variable and the independent variable. So it satisfies these two properties here. So this is just an explanation of why this confounding is being caused. But one good thing about GWAS is that in normal epidemiological studies, if you are looking at smoking versus lung cancer, right? So if somebody asks, is alcohol related to lung cancer? If we go and do a sampling, we might get a misleading result, because alcoholics will also tend to be smokers. So I will get an association with lung cancer which may not be biologically relevant, right? So epidemiological studies confounding is rampant and lot of complexity is there. But in genetics, because genes are like sort of randomly assigned at birth, you don't have too many confounders, potential confounders. It is just this ethnicity or population substrata, because allele frequencies vary. So that is the only thing that you need to worry about. You don't need to worry about age, confounding due to age or confounding due to smoking or other kinds of things. So confounding due to smoking is still possible, because smoking is genetically controlled, smoking behavior can be genetically controlled, but other lifestyle factors which are choice kind of things, you can mostly ignore. Age, gender, this kind of confounding is very common in epidemiology studies. But still in genetic studies it is a good idea to adjust for age, gender, these things, not because of confounding, but because of other reasons, that there may be heterogeneity of disease across the different age groups or gender groups. Okay. Okay. So relatedness is slightly different thing from population stratification. So relatedness, when you have collected some samples and they are genetically related more than, so anybody is related if you go back centuries, but if they are more closer than that, then that's relationship. And the thing about linkage analysis is that where the relatedness is known and modeled, so you know the exact structure of relationship and it is recent, and in case of linkage it is useful because the power in linkage is coming from the meiotic crossovers. Okay. In association, what happens is that we tend to assume that these are independent samples, we are doing regression and thing, so relatedness is assumed to be not there, but so it is unknown, distant, and if it is present, it is a nuisance thing. We are not interested in the relationship per se, but it might cause problems to our method, so we want to control for the relatedness. Okay, so when what is cryptic relatedness? We don't know exact relationship stuck between them, but there is a relationship. So this violates assumptions of many of the methods. So how do we tackle this problem? We use what are called linear mixed models to account for unknown relationship structure, where the relationship structure is estimated from the genome because it is unknown. Okay. So we will come to the linear mixed model thing because that is very important for plant and animal GWAS. But suppose we have adjusted for our confounding variable, that is population stratification, and we have also taken care of relatedness using some model. Then what we get at the end of a GWAS is that we loop through all the SNP markers or whatever markers and get p-values for each of the significance of those significance of genetic association of those loci. Now the first thing to do after a GWAS is to look at this what is called a QQ plot of the p-values, because this tells us whether the GWAS went fine or there was some problem. So this is a kind of QQ plot which will give a sort of red flag that there may be some issue because the p-values are deviating from the null distribution. Quite a few of the p-values are deviating from the null distribution. So the idea is that if there is population stratification, many SNPs in the genome are associated. So what will happen is that this deviation will start very early, whereas if you have adjusted for all confounding and everything, most of the SNPs in the genome are null, so most of the QQ plot will be close to the diagonal, and then towards the tail you will see some deviation from the diagonal because there are some truths or some really associated SNPs in the genome. So it cannot be, if it is perfectly along the diagonal, then that's bad news. That means your trait is probably not genetic, you did not find anything, maybe your sample size was too small for you to get significant p-values. Okay. So that is the idea, and actually one of the earliest methods of correcting for this population stratification problem was just taking this QQ plot, looking at its median, and then just scaling it down so that the median becomes equal to this one in the diagonal. Okay, so genomic control is a old and primitive method for controlling population stratification.
Is it drawn for only binary traits or quantitative traits?
Any trait. At the end you will get p-values for each SNP. So at that point the trait doesn't matter. So it's just you are trying to see whether your distribution of p-values across the genome is completely null. If it is completely null, it will be all along the diagonal. If it is highly inflated, then you will see deviation from this diagonal line very early. Okay, it can also go like this. If it is correct, then what you would generally tend to see is that it will go along the diagonal to a certain point. There will be some inflation in the tail. So maybe 10% of the SNPs in the genome are showing actual signals, so it will go like that. Okay. Okay, so how this is the method that came which sort of solved this principal population stratification problem is this by this famous paper 2006 Price et al. It is called eigenstat method. So what it does is it does a principal component analysis of the genome wide data, and at the end you are able to capture who is from where, like the population strata, based on the top principal components. So in this case, that you can see that top two principal components of the genome have been used to plot the individuals, and then so you pretend you don't know who is from where, you draw the points and then you color them. So first principal component 1, principal component 2, and then you color them according to the knowledge that you know. Okay, these are Americans, these are the European, British, etc. So and then that thing has just been rotated. Here you see in this one, it looks almost identical to the map of Europe. So this is Spain and this pink inside that is Portugal. So you can see it corresponds very well to the map of Europe. So here it shows that using PC1 and PC2 you can map the geographical location of the people just from the genomic data. So they gave the idea that now because we don't know who is from where, we can use these principal components instead as our covariates to adjust them in our model, and that will take care of the confounding. Another very famous method for capturing population structure from whole genome data is structure. So as you can see, this came before eigenstat, but this was not originally meant for GWAS. But anyway, you can actually extract. So what this does is it starts with guess, like I guess there are 2 populations, then I will guess there are 3 populations, then I will guess there are 4 populations. That way at some point there will be an optimum, I stop. So here I think whatever the number of distinct colors, that's the number of populations, and every person's genome is painted by these colors according to, so this person is 100% from the red population, whereas this person here is a certain person from the blue population, certain person from the green population, etc. So and they have been, the samples have been ordered in a way so that the red ones all cluster to the left and these ones also cluster to the right, etc. So this is a structure, it's called structure plot. It's very interesting. So models individuals as admixed mosaics of some ancestral population. So it will not, unlike PCA, which we are trying to say this is a cluster, this is a cluster, this is a cluster. Here it's possible that the person is half African and half American. Okay. Yeah, so this picture is from soybean. Okay, so what are the steps in a GWAS? First you need to get the genotypes from the machine, which will come as allele coding, and then you need to convert them into numeric coding, like here homozygotes are coded as 0, heterozygous as 1, and the other homozygote, minor homozygote as 2. Generally this is the preferred numerical coding. And then you need to run a sequence of quality control steps. So what are these quality control steps? Firstly, if there are samples which have a high number of missing genotypes, there is probably something wrong with that sample, the library preparation or whatever you call it. So those samples are dropped. So this 95% is just a convention, you can use other 90% or whatever. And then remove SNPs with too many missing samples. That means you look the other way, instead of looking at samples, look at a SNP and see how many individuals have genotypes at that SNP. So it might happen that 50% of the individuals, the genotype at that SNP is missing. So what do you conclude? That in your chip at that location there was some problem. So most of it came as NA. So those kinds of SNPs need to be dropped. Okay. So basic missing data filtering is done with these two steps, and then we need to remove SNPs which have low minor allele frequency. Why? Because the GWAS study design is underpowered for rare variants. If your sample maybe only 1% people have that different genotype, everybody else is homozygous. So that's a rare variant, and rare variant the power is close to 0. So why we should waste power, I mean, on such SNPs which don't have any power? So they should be thrown out preferably, and also they will give wrong p-values, because some of the normality and all assumptions that are made in this linear model, p-value and things like that, they don't work very well if you have this kind of rare variance. So that's why these are removed. Low power, and also assumptions are violated. Same for here, SNPs violating Hardy-Weinberg equilibrium, because they also cause the statistical method that is used for genetic association, that may in some way, like additive model, may assume Hardy-Weinberg equilibrium. So better to filter out those SNPs that violate HWE. Okay. So after doing those things, we inspect the population structure using principal component analysis or multidimensional scaling, and we remove. Sometimes what we will find is an outlying sample. That means most of my samples are from North India, and one sample does not map anywhere in India. So that is an outlier kind of sample. So better to remove that. And finally, we need to run GWAS adjusted for population structure. That can be using PCA, using MDS, or using structure. So, oops, I'm sorry. Any questions? Well, for the purposes of this thing, you assume MDS is same as PCA. So the idea is that MDS multidimensional scaling is nothing. You have a distance matrix between set of people, let's say, and that distance matrix may have come from anything, okay, in whatever way. And with MDS is a way of reducing that matrix into one column or two columns or three columns of variables, which will have, these three variables will then have similar distance as that distance matrix. Okay, so it is a way of visualizing that distance matrix. So you can say, suppose you do MDS of dimension 2, then instead of PC1, PC2, you will have MDS1, MDS2, and that will be a way of visualizing the distances here. Okay, so in case that distance matrix is the usual distance matrix like Euclidean distance, MDS1, MDS2 is nothing but same as PC1 and PC2. So you will find some people have done MDS, in their plot they are showing PC1 and PC2. So there is no difference for this purpose. Okay. So one minor subtle difference I will say is that you doesn't matter what you choose 10 principal components, your first principal component will be the same if you choose to calculate 5 principal components. Okay, the first principal component is the same. MDS on the other hand, if you choose to calculate one component, it will just give one variable. If you choose to calculate 3, it will give the 3 best variables which approximate that distance matrix. So depending on how many components you choose in MDS, you will get a different decomposition. Okay, now if there are no questions so far, I will proceed to the statistics of analysis of the thing. Any doubts so far? Okay, so as I was saying that in whenever there is significant amount of structure or population stratification relatedness, the preferred model is the linear mixed model. What is this model? You have the trait, you model it as a function of fixed variables, which can be, things like age, gender in case of humans, and also genotype SNPs, the SNP under consideration. So you are doing GWAS SNP by SNP. So there may be one of these variables may be that SNP, the others may be age, gender, those things. In case of plant, you could think of covariates like how much of fertilizer you have given and things like that, and the Y will be the trait of interest like yield of the plant. And okay, so here I'm sorry, so they have actually separated the SNP genotype in this variable. So these are all fixed effects for corresponding to the covariates. Beta is the fixed effect corresponding to the SNP genotype, and ZU is a random effect thing, which is sort of capturing the effect of the rest of the genome. Okay, so other than this SNP, it will capture the effect of the rest of the genome, or which indirectly gives you the relatedness or the structure information. Okay, and the epsilon is the noise which is not explained by the genome.
So can we skip the X part by including adjusted phenotype, taking adjusted phenotype as Y and then skipping this X alpha?
You can do, that is not the best way, that will be have less power than doing the whole thing at once. Okay, so it is like saying that so y equal to a plus bx plus cz. Okay, so suppose x is your IQ, z is how much you have studied, and y is the marks that you have got. So what you are saying is that, can I first look at just this, subtract the thing, take the error here, and then look at it with only with z, right? So you are saying this, right? I will calculate the error from the first regression, and then that error I will regress only on the other variable.
So not error but the adjusted phenotype.
Adjusted phenotype is nothing but the fitted, this is the fitted value of the thing, and then original minus the fitted thing. So that's the error, residual. So then you do, okay. So sequentially regressing the variables is not same as doing multiple regression. That means two at the same time. There is a lot of loss of power in that. It will give you something, I mean, you will not get probably wrong type 1 error, that means false positives, but in terms of power it will be much lower. Okay. So okay, so X is the matrix of fixed design matrix for fixed effects, Z is the design matrix for random effects. Sometimes it will be identity matrix, that means for each individual you have a different random effect, U1, U2, etc. This is a random effect explained by the genome for each individual, and okay, so this is what it says is that the U's are distributed as normal distribution with zero mean, and variance, genetic variance is sigma squared G, and K is the kinship matrix, or also called the genetic relatedness matrix between these individuals or samples. Okay, so I think the model is clear. Now can somebody tell me, suppose I have two monozygotic twins, identical twins in the sample. So what will this look like for those two? So Y1 is the, oh, it's not that. So Y1 is the first twin and Y2 is the second twin. Okay. So what will the thing look like for them? So x1 prime alpha, x2 prime alpha, okay. So whatever, I'll just write x1 into alpha x1, and beta times G, the genotype is same for both of them, right? And what about this? Z1 into U, okay. So forget about Z, suppose it's just U. Do you think these two will be equal? Yes. Why? Yeah, because of this kinship coefficient between them is I think 1 or 2, whatever. So perfect correlation, so their random effects will be the same. So this will automatically force their trait values to be the same. Okay, so if people are more related, this model will force their trait values to be the same. If people are completely unrelated, then you can have U1 equal to +3 and U2 equal to -3. Okay, so that is possible because these are unrelated. So their trait values may also be completely different. So the random effect in this way controls the relatedness between, and relatedness can be through actual relationship, or because they belong to the same population strata, so their allele frequencies are similar, so that way their trait values would be similar. Okay. This is a recommendation in this paper of when to use which method. So genomic control, as I said, is a very early rudimentary method which only takes care of little bit of population stratification and very little bit of relatedness. But if you have lot of relatedness, not necessarily population strata, you have to use mixed models like linear mixed models. If you have lot of population stratification but not necessarily relatedness, you can use structured association like structure. And when this is the extreme case where you have both lot of relatedness and both of lot of population stratification, you should be using a linear mixed model with actually population structure. So if you run the structure program, there is this admixture kind of information. So that should be used as a covariate there. Okay, eigenstat works in this sort of moderate kind of scenario where you don't have too much of structure or too much of population stratification. Okay, now once the QQ plot looks fine, and that means population stratification has been adjusted for, we want to visualize what are the results we have got. We do a Manhattan plot, and all of you must have seen Manhattan plots. So what is this? X-axis is base position, sort of cumulative base position along the genome. So that means where chromosome 1 base position ends, you just add from there for the second chromosome. This way you construct the x-axis, and y-axis is minus log 10 p-values. And in case of humans, you have, because based on the density of the chips, it is a convention to use 5 into 10 to the power minus 8 is a p-value threshold. So you need to do what is called multiple testing correction. So you need to adjust put a stringent genome-wide threshold. Now in case of plants and animals, I don't know what are the conventions, and it may be different for different species also, but FDR is a one good way to go which works anywhere. So instead of putting this kind of a hard threshold, you can try to do an FDR correction. Okay, and this kind of Manhattan plot can not only be done for a single trait, it can be done for many traits together. This is an example where a lot of metabolomic traits were analyzed in a GWAS, okay, in this paper. And there are other things. The gene expression can be considered as a quantitative trait, metabolomics measurements as quantitative traits, anything, not necessarily just. Yes. Down. This is not FDR correction. I think this is just the same thing that is like without colors. So overall idea you get from here, and then here you get to look at which particular metabolite is but you have to check the paper to actually see what it is. Okay, so linear mixed model, we have seen a very complicated model. How to fit that is the question. So it is one way to fit that is using exact maximum likelihood or restricted maximum likelihood for each SNP, one at a time. This is computationally very slow, but this is the exact, there are no approximation here, so this is the best, most powerful thing. There are these software for fitting the exact ML or ML for each SNP, and recently this fast LMM has been proposed, which is very fast. It does exact thing, but it has a limitation in that it works only when you have more individuals than SNPs. Okay, so how many of you work with GWAS data or are going to work? Anybody else? So anybody who has more samples than SNPs, raise your hand. No. Okay. So that is the scenario where fast LMM works, is millions of samples. So it is now, I don't know whether Microsoft people originally made this, but now it is available from Microsoft website. They basically maintain this. Okay, so what is more popular is methods like these, which sort of say that we don't need to do the variance components or heritability estimation for each and every SNP separately. We can do it once for the entire genome and then fix that parameter and just do SNP by SNP tests, which are like least square kind of test. So this is called so MMax does a score test for each SNP. P3D is available in Tassel, which does likelihood ratio test. And there is another method which is simple linear regression for each SNP, but it's very bad, as I will show you. Compressed MLM is a method proposed by the people who have written Tassel, and they have shown it works well in terms of power. So the idea is that instead of saying that there are n many random effects, U1, U2, UN, you put individuals who are very close together in terms of their kinship, like these monozygotic twins will come to the same cluster, so their random effects will be the same, one random effect U1. So this way it will reduce the computation time and also sort of improve power, because it is unlikely that very closely related people need different random effects, because they will not come from different strata. Okay. Okay, this is a comparison of power between these methods. This appeared in the MMax paper by Zhou and Stephens. So there they have shown that grammar has very low power, and this MMax, although it's an approximation, it is in most cases almost as powerful as the real, I mean the EMA, that means the exact ml or ml. And in terms of speed, if you can see here that Mx is actually quite fast. So it's as good as the best thing, that means in terms of power as good as these three. In terms of computation, it is much faster than EMA and also faster than FastLMM actually. So Mx is a good method to use. If you use Tassel, you will find P3D implemented there. If you use Gapit, it has both P3D and Mx implemented. Okay. So coming back to the model, this is the model. Here there is no SNP, but if it is there, you can assume it is within this fixed effect design matrix. So how does this thing work? The first thing to do is basically to estimate the random effects, but suppose the random effects are known. Suppose you know exactly this variance covariance matrix, then that means you know this G, which is the variance covariance matrix of U. Then, any questions? Okay. So first you think of this whole thing as your error for which you know the exact covariance structure. Then it is just like a simple GLS model. So this is the least square estimate. Okay. And on the other way, suppose you know the beta hat. Okay, this is known. Then you can just subtract y minus x beta hat, and then this is like a normal problem, normal mixture problem. So you can take the posterior distribution and expectation of that. So this is the base estimate. Okay, so this is the base linear unbiased predictor, and these two solutions you can put in terms of this linear equation, which is called the mixed model equation. So solving this mixed model equation is same as solving these two equations separately, and so this fitting proceeds in this way that first you need to estimate the variance components, and then you fit this mixed model, solve this mixed model equations, and sometimes you need to iterate between these two steps. I know not everybody is from statistics background, but if you have any specific question on this, please let me know. Which one?
Blues and clubs.
So for a fixed effect like beta, you can give what is called base linear unbiased estimate. Okay, that is the sort of MLE you can say. Okay, also it's also the minimum variance unbiased estimate. For a random effect, you can get a posterior distribution. Okay, so actually if you assume that y given u is normal, right, and u is a priori normal, because we have assumed that u to be a normally distributed random effect. So y given u is normal, so by Bayes theorem, by conjugacy of normal, posterior distribution is also normal, so you can get a posterior, and the expectation of that distribution is this. So Bayes linear unbiased estimator, Bayes linear unbiased predictor is nothing but the Bayes estimate, that means mean of the posterior distribution. Okay, so in any mixed model you can get a blue of the fixed effects and the bloop of the random effects. So now this one here shows you how you do genomic prediction using the Bayes linear unbiased predictor. So GWAS is one thing where you do SNP by SNP association, but using the same model you can, so forget about a single SNP at a time. If you just fit this genome wide, you can get the Bayes linear unbiased predictor, and the way it works is that you have some animals here, but you don't have the trait values for all of them. You have the trait values for these three. Okay, So these are your reference kind of sample, you can say, and you want to get the predicted values for those for whom you don't know the trait values. So those are your like inference sample. Okay, and now the relationship structure between these animals is given here. You know that. Okay, so again this is the model here. You have 5 random effects corresponding to each of the 5 animals. These are the 2 fixed effects corresponding to herd. I mean, if they all belong to the same herd, then this herd covariate will not be there. But in this case, they have assumed there is an environmental effect of the herd, so you have a fixed effect corresponding to herd. And once you fit this linear mixed effect model, you will get a bloop of the U1, U2, U3, U4, U5. So you can use those 2 missing 4 and 5 whose trait values you don't know. You can use the bloop of those 2 as your predicted trait value for those 2. But I mean, without going to the model and how intuitively where is the information coming from? That means I know the trait values of 1, 2 and 3. How am I predicting the trait value of 4 and 5? Where is the information coming from? So because suppose this trait is a polygenic trait, I mean across the genome there are many loci that are controlling the trait. Okay, so the kinship matrix will sort of capture overall genomic similarity between an animal for whom you don't know the trait value and the animal for whom you know the trait value. So if the closest animal for which you know the trait value will sort of give you some information about what should be this animal's trait value. Okay, and mixed model equations that I showed before, you can have more things there. Instead of 1 variance component, there are 2 variance components over here. Y. So here there is a linear model with fixed effect of sex and random effects of common environment related to each litter. So WC is random effects for common environment, and these are the breeding values, the remaining thing. So in this case there is 3 things in the mixed model equation, but the concept is the same. So first we need to estimate the 2 variance components, that means these 2 ratios. Once that is there, you just solve the mixed model equations and you will get the breeding values or predicted values. And these random effects are estimated generally using restricted maximum likelihood. Any questions so far? Okay. So what is genotype imputation? Any idea? I am sure some of you have used it. What does it do? Yes. So the idea is that we started with this in human GWAS, started with this 500,000 chip, 500,000 SNPs in a chip, and then it was found through the HapMap project and then 1000 Genomes project that SNPs are very closely related to each other, LD structure etc. is known. So we can genotype fewer markers and from that we can actually fill in the rest of the SNPs. So that is imputation. It can also be used to fill in missing data here and there, like one person is missing here or one SNP is missing there. That can also be done, but the original purpose of imputation was that this SNP is completely absent from the chip, nobody is genotyped, but I know how this SNP relates to the neighboring SNPs which are genotyped actually. So from that I can predict what is the genotype of this SNP for every person, okay. And that LD information generally comes from external reference panel like HapMap or 1000 Genomes project, but without reference panel also it is possible to do, okay. So there is a, yeah, so there is some software for population based data where there is not a lot of relatedness is not there. These are the two very famous software. For animal data, I have seen people use FImpute, and for plants there is also FImpute, and there is something called Fillin, FILLIN. Okay, so this is what trying to explain what is imputation and why it was done. So these are the sparse SNPs that you have on your chip, okay. Now the idea is that if I am able to fill in the genotypes that are missing on the chip, it might happen in some cases that an imputed SNP has a much stronger p-value than the actually observed SNPs. Why? Because that imputed SNP happened to be much closer to the causal variant, or actually it is the causal variant, okay. So how is this done? It's like this. So you have this is the genotype data. So first step is what is called haplotyping. So for using several methods like EM algorithm, HMM, etc., you try to guess what is the most likely haplotypes for this person or this sample. This is a genotype, right? See 0, 1, 2. So this is a genotype. From there we are getting 2 haplotypes. So this 0 plus 1 is 1, 1 plus 1 is 2, okay. So 2 haplotypes, splitting into 2 haplotypes, and then we look at the reference sample, reference panel, this say 1000 genomes or HapMap it comes from, and we see that these positions where there are question mark, that means unknown genotype, so unknown haplotype. So if it is red, so this is the red, look at the red haplotype here. So what happens in the red haplotype over here? If you have a 0 here and you have a 0, 1 here, look here, 0 here and 0, 1 there. So you know exactly what these should be. So in the reference panel you have the most likely haplotypes. So you fill in these question marks by these zeros and ones, okay. So accordingly now you can predict what would be the genotype here, because whatever I have got from the red haplotype here, whatever I have got from the yellow haplotype here, those should be added up to get the genotype here, okay. So so this is the idea. Each sample is phased and the haplotypes are modeled as a mosaic of those in the haplotype reference panel. So this is a mosaic, that is a mixed thing of these haplotypes, and once you have constructed that mosaic, now you just add up the whatever is in the red one or whatever is in the blue one, and you will get the genotype over here. So that is imputation. Once you have imputed, now it's a complete matrix. Now you can do your GWAS and look at what p-values you get, and it is sometimes you might get lucky and your imputed SNP will give much stronger p-value than the original SNPs. So imputation is used for that purpose, but it is also used for what is called fine mapping. That means I know my GWAS has given me strong evidence that there is a SNP or locus in this area which is controlling the trait. Now I'll dense impute on dense panel and I'll try to locate what is the causal variant. That is fine mapping. So imputation can also be used for fine mapping and visualization of the region that you have found through a GWAS. This is what is called haploview LD plot. This is also another thing which is used for fine mapping. How? So suppose you have found a GWAS SNP strong p-value over here, another one strong p-value over here. So you know that these are in the same LD block, so they are all correlated to each other, so these are not two separate signals. But if you find one p-value of strong over here and another p-value strong over here, you know that these are two independent signals and you need to map them separately. Okay. So how to do fine mapping? Number one, imputation. Number two is by what is called sequential conditional analysis. That means you pick the topmost one, condition on that, look for the next topmost one, and go like that. And there are Bayesian methods which give you posterior probabilities of causality. Okay. So you have done your GWAS, found a region, mapped the causal variant. Is that the end of the story? That is the end of the story as far as your genome to phenome mapping is concerned. But you have no idea how this happened or how this SNP actually affected that phenotype until and unless you look at the intermediate steps. Okay. So and that is the bread and butter of integrative genomics, which is still a nascent subject, and there is a lot of thing to do in this area. Okay. Any questions so far? So Tassel is a software developed by a Maze Genetics group, and it is one of the popular software for GWAS in plants mostly. Somehow I'm not aware of much differences between plant genetics, animal genetics. Why it is not used that much in the animal genetics sector, I have no idea. Humans I can understand, but it's like people work in different communities. Plants only read plant papers, and animal genetics only read animal papers. So this is not used that much in animal genetics. So it's a Java based software, and most of the things you can do by just load the file and click and things like that. But it also has an underlying command line interface where you can build pipelines for Tassel, but the graphical interface is more popular, I guess. So Plink is developed for human GWAS, and it's still probably the most popular software for human GWAS and also in animals. So actually, one good thing about Plink is it is very well maintained. So generally these software are made by somebody working on a particular GWAS at some point, like a PhD scholar or postdoc scholar, and what happens after that is once they go to something else, nobody maintains that and it goes. But this has been consistently maintained for a long time, and now a new version Plink 2 is also being developed parallelly, which supports more VCF file, sequence data, things like that. Plink is very good for initial quality control of your data, like filtering SNPs and things like that. But when you go to the end of it, that means actual GWAS with sophisticated statistical models, Plink does not have. Plink has very basic linear regression, and even the logistic regression is not that good. Okay. So these are some of the paper references which I mentioned along the way. So I'll stop with the theoretical thing there. The other thing that is not mentioned over here is GAPIT. GAPIT is an R package, not an R package actually. Again, it is a R code that is maintained by somebody, not very well maintained, but it has a lot of functionality. So we'll try with the GAPIT dataset in the next session. With the GAPIT dataset, we'll try to run GAPIT as well as we'll try to run Plink and Tassel, and just try to load the data and play around with it, because ultimately in this kind of thing you cannot rely on one software, because GAPIT might have some bug, it might give some wrong result, so you should always cross check with some other software, Plink or Tassel or something.
So which one is better for association using SNPs or haplotypes?
So I did not talk about haplotype analysis. So haplotype analysis actually you do SNP by which are not in very strong LD. So say 0.3, not very strong LD, okay, similar in the same sort of region. So then you should think about haplotype analysis. That means that these two together might be doing something. So it could be that there is a causal variant somewhere in between, and if you do an haplotype analysis, you will be actually capturing this causal variant well, okay. Or it could be something else. That means this is a promoter variant, this is an enhancer variant, and these two interact in some way. So you don't know. So you can do those only if you find some reason to suspect that there may be more variants, more than one variant that is associated in this region, okay. So haplotype analysis is saying that the A allele at this SNP and the B allele at this SNP, when they are present together, that is affecting the trait in some way, okay.
So would you be focusing on downstream processes after GWAS as well, like how to locate the genes for which SNPs have come out to be significant, and then how to find the pathways? Would you be focusing on that?
I am done with my talk. Now we will be doing practicals, okay. Practical means simple, it's just to try and run a GWAS kind of thing, okay. But if regarding your question, you are interested in downstream, how to map genes to
Genes and then start finding pathways.
so there are very good, as soon as you are reduced to a SNP list or a gene list, there are many good packages in R bioconductor to do all kinds of annotations like KEGG and gene ontology and things like that. So we will not be focusing on that, because the idea today is to go from basic genotyping data to your associated SNPs, which you can then map to genes and go on to pathways.
So if I want to do for my research work, which softwares can I use?
So bioconductor, so you use bioconductor variant annotation, okay. Variant annotation that with that you can map your SNPs to genes, and you can use these KEGG.db, geneontology.db or reactome.db, whatever pathway database you want. That package you need to load, and there is a package called GSEA, which is gene set enrichment analysis, which does pathway analysis kind of thing. Other than that, you can go take your gene list and go to directly to what is this called? David, I think. Okay, David is a web tool which does pathway analysis. And in David, you need to go and select which database you want, KEGG or BioCarta and things like that. So it will do the pathway analysis for you. It's just click things. R is the harder way to do it, but you have more flexibility with things. And you can do cytoscape either directly or through R. So R has package for cytoscape kind of, so network analysis, all those things. Okay, any other questions? GPlink is a graphical way of running Plink, which has a limited set of features. So Plink itself is so easy that the graphical interface is not that popular. Okay.
So this module is
So multi-marker models, if you go to Plink, you will find some option, I think, but you need to look at that literature, particularly, I mean, the gene-based. So just search for gene-based test. Okay, I forgot the name of the software now. I forget no, I don't. Once when I recall, I'll tell you. So multi-marker means it is usually you would look at something like a gene or kind of an entity. So the methods are similar for gene-based test and multi-marker, whatever it is. There are standalone software and there are also R packages for that. But I'll let you know. Just if I forget, let me know. Okay. Shall we proceed to practical? So first of all, do you have गैपिट इंस्टॉल हुआ है? तो सर्वर में जाना पड़ेगा। इंस्टॉल विंडोज में
वो आप एक बार कोड यहाँ शो कर दीजिए।
हाँ, कौन सा?
नहीं तो मैं मेल करा दूँ जो आप भेजे थे ना, उसमें सारा मेल हो जाएगा ना, उसको कॉपी पेस्ट कर देंगे तो आर में तो इंस्टॉल हो जाएगा वो। तो वही पैकेज वगैरह ना जो।
हाँ, ठीक है। Actually, what you can do is I'll give a पेन ड्राइव में चीज़ है, मेल कर देंगे। Let them download. I'm just showing the website. Let's let them download.
उसी से भी तो कॉपी।
हाँ। Okay, so first let me show you the Plink and this thing quickly. So Plink, if you just type Plink genetics in Google, it will take you to this website. Okay. Oh. Which one? Which one? वही चला गया क्या? चल रहा है,
वाईफाई को कनेक्ट नहीं सर
कोशिश कर रहा हूँ, HP वाला तो नहीं आ रहा है।
केबल वाला कीजिए
हाँ, केबल,
केबल
केबिन, ये
आईटी केबल वाईफाई मैं पूछ लेता हूँ एक बार पूछ के
you guys are working on Wi-Fi or is Wi-Fi or net? Ethernet, na? Oh, that's good. Okay, and as soon as I get something, it can come. हाँ, केबल होगा?
एचपीसी करके पूछे देखिए नीचे है क्या एचपीसी
इसमें होगा क्या?
और यहाँ का केबल
इस ये कनेक्टेड है?
हाँ इसमें नेट कनेक्टेड है इसमें नहीं
देख लो।
और आपको चेंज करना पड़ेगा उसको बंद करना पड़ेगा
इसमें R भी है, चलेगा।
सारा कुछ है इसमें
ये खोल दिया।
तो इसमें
Okay, so resources available for download, teaching materials and example data set. Okay, so I think this is example one is easier. Just download the example.zip, save it somewhere. Where did it go, by the way? I'm not. Hey, where is Windows Explorer? Windows E. ये sir. Okay, so it's difficult. Wait. That's fine. That's fine.
First, we use PDIC now, sir.
Uh-huh.
First, we have to access PDIC now for all the systems.
Yes, but
This is command line, ping command line.
this is
In
plain
Windows.
command line, yes, and they have it right in the desktop. Okay, so on your in your downloads there is a folder called Plink Win64. Everybody has this. In the downloads there is a folder called Plink Win64. Go to that folder, you will find these files, okay, and you need to copy this example folder which you get from this example.zip into the Plink folder. So downloads, okay, downloads you sort by name. There is a something called Plink, alphabetically you go Plink Win64 and then go inside that folder. There you will find these are the files, okay, and what I am saying is we need to copy the example.zip, extract the example.zip and copy here. वो तो डाउनलोडेड होना चाहिए. और ये है ना,
वो हमने उसको डाउनलोड किया था अपने सिस्टम में सपोर्ट करने के लिए.
ये हाँ यही चाहिए, यही वाला फोल्डर चाहिए. ओके, यू डोंट नीड टू मच, जस्ट नीड दिस फोल्डर. इसको कॉपी करिए.
वो सर फोल्डर में सही जगह रखा हुआ है ये.
अच्छा, तो एग्जांपल, ये एग्जांपल वाला फोल्डर
एग्जांपल हमने अलग से डाउनलोड किया था ना, वो वाला है सर.
हाँ, उसको इसके अंदर डालिए, यहाँ पे डालिए. यस.
It's coming like a zip file. Whatever I have downloaded is coming like a zip file.
Yeah, so just double click or, oh no, no, is that how you're getting it?
Link 2.
Pink 2, no, I don't need pink, just pink 1. So just go to Plink Genetics and go to the first one, Harvard thing.
एग्जाम्पल को हमने किया तो उसको ये एग्जाम्पल वाला फाइल है, एग्जाम्पल वाला फोल्डर इसमें ये आ रहा है। अब इसमें से क्या लेना क्या है सर? एग्जाम्पल की फाइल में जो डाउनलोड की
ये एग्जाम्पल वाला है?
हाँ ये एग्जाम्पल वाला है।
इसके अंदर लिंक है?
इसके अंदर लिंक है, हाँ एग्जाम्पल
मेरा तो okay, so let me repeat from before. Now it's easier. Okay, so you have gone to the resources, and then you have teaching materials, example.zip. Download this, and then you have to extract this folder. It will come as an example folder. In that example folder, Plink is already there, so you don't need the other Plink that you have downloaded. Okay. Sorry.
Which is not visible from the back side.
Oh, I see.
Now is it visible from back side? Not visible? Now? Oh.
No, it will not. It will not launch. Just
Now visible? Is it visible?
that's fine. This is too large. Too large.
Because it's not visible from the outside, so they are not following you.
So download example.zip, and after that you need to extract the example folder. Inside the example folder you will find all the input files. You will also find the Plink executable is there. So we can only work inside this folder. You don't need anything else. Just need to download the example.zip and extract into example folder. Okay. So open nahi hoga. So don't double click on the plink.exe, it will not work. So let everybody first get this folder, then I'll tell you the next step. Okay, just go inside the example. Go, yeah. There you have everything. Okay. So anybody who has not got this example folder, anybody who is not able to get up to the example folder? Got there? Okay. Okay, so press Windows and then write CMD, press enter. Press Windows key, write CMD, press enter. Got it.
What we have to do next?
Which folder is this? Oh, you have saved it as Plink example, right? Yeah. So just see all the files are there. Yes. So here if you write Plink, now it is working. Okay. Now Plink is running. So you can follow the tutorial there. Yes.
सर ये तो डाउनलोड कर लिए थे पहले सर इसके बाद
हाँ, इसके बाद CMD जाके यहाँ पे जाके
हाँ
CD करके वो फोल्डर पूरा कॉपी करिए यहाँ से, यहाँ पे कॉपी करिए CD,
After Blink, what we need to type? Change the directory.
CD within double quotes, paste, space, CD space,
सर 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 सर
हाँ
लॉन्च नहीं हो रहा है क्या ये?
हाँ वो लॉन्च नहीं होगा. So this is the command. So we press Windows key and press CMD. After that you need to write down CD space double quotes में paste the folder, entire folder path. So here, from here you take, just click here, you will get the entire folder path, copy it, right click, copy, okay. In the CMD you write CD space within double quotes paste, right click, paste the entire folder path, and then double quote close, enter, so that you enter that folder. Everybody done till that? Okay. Anybody get stuck, please let me know. Where are you getting stuck?
Sir, I have created one separate folder, a Blink example, in that the zip file is there. But now when I put it here, now it is telling Blink is not recognized as internet.
No, you're fine. So you are already in that folder. You have saved it as Plink. Sorry, what is Plink example? This one.
This one.
You have not yet extracted it. That is the problem. So select all. Where to extract is asking. Just give a name.
Okay, sir.
Some maybe you can keep in desktop or somewhere. Just keeps folder name has to be.
Okay, sir.
Just no, don't copy what I have written there. You copy from here. Yeah, so just check. You have all the files here. Yeah, you have.
So the same thing can be done for this Mac.
Yeah. Mac, you need to.
So software will be different for this.
I don't think so. Did you download it? The example that's what's going on. It should be same. You just need to open terminal. Have you opened terminal ever on a Mac? It's usually hidden away, but you have the same command line thing. Right click is control click, command click. So here, you go here, there is this applications utilities. Not applications, it is in
documents. So
applications.
it has to be saved here.
Search terminal. Or there is a easier way, I think. You can search system wide. There is a search. No? For what? There is a system wide search somewhere here. Yeah, spotlight search, right? Terminal. Just need to open this. Wherever it is downloaded, you just need to go there. Download is complete.
No, sir, not yet.
Not yet. So you will need to go to that folder. The zip file you need to unzip, like unzip file name and then cd into that folder. cd. Okay, so
ब्रेक करना है ताकि के बाद आधा घंटा कर लेंगे जो भी होगा इसको खत्म करते हैं.
ठीक है.
पहले 2 मिनट का ब्रेक करेंगे.
हाँ, let everybody get till here. अभी बैक करना चाहते हो exactly right now?
अभी आप कोई बीच में तो नहीं कुछ
नहीं, ठीक है, ठीक है.
तो let us break for the launch. So अभी कोई continue कर सकते हैं.
Okay, so so I'll just say in this example folder there is a file called commandlist.txt. Okay, if you right click on it, do you get this option edit with notepad plus? Yeah, so then you open that. This is not, because if you open it in notepad you will not be able to understand what I think. So these are all the commands, part 1, part 2, etc. for Plink that will run on this data set. So you can just copy one command at a time and paste in the command window here. Okay, so you will run, it will give some output, and in terms of interpretation of this output you have to go back it's easiest to interpret yourself. So anyway, so the output is usually self-explanatory. So it will in Plink. So this first step, whatever it happened, it's telling you everything over here, what it has done. Okay, but we'll come back and explain some of these commands. But basically, to run this tutorial, all you need to do is go to this command list thing. Command list we have opened in Notepad++. Choose one command at a time and paste it here. Okay. So that's about it. So we'll come back after lunch and